Awesome. Welcome everybody to another Ask the Expert Talk. I'll be your host today, Andrew Long of the Sean and Parker Center for Allergy and Asthma Research, where today we will be chatting about novel therapies of food allergies. I hope to give you a little insight on where we're at with treatment and where we're headed. We'll see what happens. So just to set up a little background, past 2 decades not looking so hot for food allergy as a whole, with not only the incidence increasing, but also severity. The presentation of food allergy varies across patients. The majority are IgE mediated, which is the classic food allergy that you usually think of with rapid onset and anaphylaxis. And as such, it's currently the focus of most clinical trials. Despite being the most common, it's definitely not the only presentation. There's both non-IgE mediated and mixed pathology out there. It's important to understand that because that dictates the way that we approach treatment. It heavily depends on the mechanisms driving the disease under the hood. So a little bit about the pathology. We won't go too far into details, but it sets up a good foundation. In the development of allergy in general, at some point the body needs to make a decision on what to do when it encounters an allergen. Antigen-presenting cells are constantly picking up neat things it finds in the body, breaks them down, and it shows them to naive T cells. T cells are what we have in our body that helps us determine how the body responds to things. And naive T cells refers to the new T cells that really haven't found their way in the world yet. When the APC presents the potential allergen to the naive T cells, the local environment and the APC direct the T cell down a path. It kind of tells it what to do. In the development of tolerance to food allergen, the APC promotes the naive T cell to differentiate into antigen-specific T regulatory cells, which we call Tregs. In the case of allergy, however, it promotes maturation into a T helper 2 type cell. Sounds helpful, but in this case, not so much. The Th2 cells promote the production of allergen-specific IgE by B cells, and the IgE are antibodies that recognize and bind the allergen next time it's encountered in the body. The IgE sits on mast cells and basophils just kind of hanging out, and then the next time the allergen rolls around, that's when the allergen binds to it. It brings multiple IgE together on the surface of the basophil and mast cell, and as that happens, causes degranulation and the reaction that we love. So we're not really going to go through these abstract paintings too much, but they sum up the previous slide a little bit. As you can see in the black boxes that I put up, you can see where IgE plays a role that we were kind of just talking about on the left side. Sticking to the mast cells awaiting future exposure to the allergen. On the right, it's kind of just a magnified view. It shows where the antigen binds the allergen. It brings multiple IgE together. And when that happens, all of this black magic happens, eventually resulting in a reaction. So this is kind of a figure that we put together for a review that we wrote on this topic a little while ago. Again, the main takeaway is not to understand the whole picture, but more so that IgE, even though we've talked about it here, it's not the whole picture. There's tons of mechanisms going on, and no single one is responsible on its own. It's really a net effect of all of these markers. IgE is on the left side, on the basophil and mast cell, the teal and purple shape. Those are all the way downstream. And so it's really the result of everything that's happened upstream. By blocking it, you get a very specific effect. But as you target markers up the stream, further up the cascade, you get a broader effect with a trade-off of higher risk for more drug-related AEs, adverse events, as you block everything downstream. It's kind of hard to see, but all of the markers in bold are currently potential targets for intervention that we're looking at in food allergy. So how do we fix food allergy? The fundamental strategy now for over 100 years has been repeated exposure to the allergen gradually escalated over time. Naive T cells are always being produced while old ones die off. In food allergy, the naive T cells are always preferentially differentiating to Th2 cells, which just perpetuates the allergy and maintains it. We definitely know by now that repeatedly exposing the body to the allergen over and over and over shifts that preference to development into Tregs. It shifts the environment and the APCs, and you can see downstream effects from inhibition of mast cells to class switching of B cells to cause them to stop pumping out more IgE. Over time, your tolerance slowly switches from Th2 dominant to Treg, and you build up your tolerance to the allergen. That being said, it's not the greatest on its own. We all know what happens when you eat something you're allergic to. I wouldn't call it awesome. To facilitate repeated exposure that we need as safely as possible, it really requires starting at very small doses of oral immunotherapy and very slow escalation to avoid hitting that level that would trigger the mast cells or basophils to activate. It can take 1 to 3 years to get up to a serving size in most protocols of a single food. Typically, due to the small volume that we can do at once, because of that limit, it's really limited to a single allergen at a time, which isn't that helpful, seeing as more and more patients are presenting with multi-food allergies. So really taking 1 to 3 years to solve a single allergen is not ideal. And sometimes the reaction rate, even at the lowest dose, when we try our best to go slow and start at a very low dose, precludes this method. So it's really back to epinephrine and avoidance. Beyond the IEEs themselves, the second major challenge that we've had with just OIT alone is the need to continue routinely eating the allergen at the tolerated dose level when you're done to maintain the desensitization. Really, we've only seen a fraction of participants able to maintain their tolerance after they stop eating it, which we term sustained unresponsiveness, or SU. In a study we actually conducted in peanut allergy 2 years ago, we saw that after desensitizing patients to tolerate 4 grams and then discontinuing it, only about 40% maintained that tolerance after 3 months, dropping to 15% after 6 to 12. Sustained unresponsiveness is really the holy grail that we're looking for in food allergy, seeing as it's, quote unquote, a cure that lasts after you're done with treatment without the need to eat the allergen on a regular basis. So given all of this, it's pretty clear that we need to improve upon just oral immunotherapy on its own. And there are 2 main strategies being studied right now in doing so. The first is using adjunct biologics to selectively, temporarily suppress part of the immune system just responsible for allergic reactions and the whole inflammatory pathway, increasing the amount of allergen the patient can tolerate before they react. It's kind of an artificial increase in their tolerance that opens up a window for much safer repeat allergen exposure while increasing the rate that we can actually escalate the dose. The higher allergen dose tolerance means that we can accommodate multi-allergen therapy. And the thought is that with the involvement of more immunologic processes, we can actually be able to increase the odds that we will achieve sustained unresponsiveness. After the allergen is built up with the biologic and the body shifts to tolerating on its own, through the repeated exposure, the biologic is discontinued. So it's not a permanent thing. It's more of a temporary window and temporary solution just to expedite the allergen and make it safer. Of course, there's limitations, as always. The limitation is that these biologics are only able to suppress or modify the immune system itself. And you still need that allergen exposure to actually retrain the naive T cells and the body and maintain the desensitization after you stop the biologic. You need your body to start retraining and take over. The second most recent strategy in development right now does not involve biologics at all. It's really the use of modified allergens where the molecule used provides repeated allergen exposure to the naive T cells without triggering any of the reactive pathways. And it's very nuanced, and you know, it's still really in development, but I think it holds the most promise right now. For example, it's, you know, there may be a molecule modified in such a way that it's not recognized by IgE, so there's no risk of a reaction, but it can be seen by naive T cells, so you're getting that repeated exposure and retraining. So we'll talk about that a little bit later. So taking everything together, the current strategies right now involve allergen exposure by the methods listed here. You know, oral, sublingual, epicutaneous, and the modified allergens we just talked about, with or without the immune modulators at the bottom here. And you'll be familiar with some of these, definitely. Many of them are approved for other disease states and atopy. It's definitely not an all-inclusive list, but these, to me, are the furthest along with the highest promise right now in food allergy. So first up, the tried and true, always loved anti-IgE agents. As we discussed earlier, IgE is what recognizes the allergen in the first place and sets off the reaction, so let's block it. By blocking it, we reduce the IgE on the basophil and mast cell, requiring more allergen consumed before enough IgE can be bound to cause a reaction. Currently, the most studied biologic class in the allergy, with omalizumab, is the most studied agent within the class. Many of you will be familiar with this agent. A number of studies here and across the globe have looked at this in food allergy. So for that point, I won't cover it too much in detail because it's been covered ad nauseum, but in summary, it's been studied both on its own and with single or multi-oral immunotherapy. It's been found to be significantly efficacious in facilitating faster OIT escalation and reducing adverse event rate and severity during exposure as expected. That being said, it isn't perfect. It definitely has some room for growth and optimization. Even though it shortens your time to reaching your maintenance dose, you know, 2 grams, 4 grams, what have you, it does not increase the proportion of patients that do eventually reach maintenance with enough time. If you go slow enough and cautious enough, with those on placebo can hit the same OIT dose as those on Xolair. It's just a matter of time and the adverse events that occur. Additionally, even though it does lower AEs, there's still persistence across patients at variable rates, with the majority of those being gastrointestinal. For some reason, it has a little bit less efficacy in blocking those. And eosinophilic esophagitis still persists in about 10% of patients. So we're finding it doesn't have as much benefit there either. The data so far does not indicate that it increases sustained unresponsiveness. And really, there's still, you know, surprisingly, a lack of consensus on the optimal dosing strategy for it in this specific disease state. Right now, we've seen quite a bit of variability in its ability to increase allergen tolerance across patients, with approximately 10% of patients actually seeing that they tolerate still only 5 milligrams or less after full dosing of omalizumab. And we're still trying to understand why. You know, perhaps those patients need a higher dose or a longer duration. We've actually just presented our findings on this in our own population, kind of looking at influencers of dose and the response at the IACI conference, and that should be available online if anybody's interested. So right now there are actually 2 main trials trying to answer these very questions. How do we best use Xolair? The first of which is Outmatch. It's the largest, and it's a phase 3 actually. It's a nationwide study sponsored by the NIH with 10 different sites across the US. Stanford is one of them. It's really looking at multiple durations of omalizumab use on its own, ranging from 16 to 52 weeks, and comparing that to omalizumab pre-treatment followed by multi-allergen OIT itself. Then assessing the efficacy as well as how the duration plays a role in this. And the third stage of the study is actually looking at transition after therapy to daily consumption of real food equivalents after stopping both the omalizumab and OIT. On the dose finding side, there is the BOOM study. It's currently gearing up to start. It's looking at assessing how to maximize the treatment speed within patient safety using weight-based dosing only. Up until now, we've always used the asthma dosing for Xolair, which is based on IgE and weight. This study is looking at just weight alone based on some preliminary data that's been published. So we're looking forward to seeing how this pans out and if we can increase the range of those participants who can benefit from the drug, because really, despite these limitations, it is a great pathway. And it's a very valuable mechanistic target. Being so limited when we block it, it's not really prone to other, full body immune suppression. IgE is very specific to food allergy itself. And so it's a nice target downstream. And given that, there's other agents outside omalizumab that are in the pipeline. We have quilizumab, which binds IgE already bound to cell surfaces rather than free IgE like omalizumab. So it may be a better option in those with very, very high IgE compared to Xolair, which means you need a high enough dose to saturate the free IgE. And so Xolair caps out at a top monthly dose of 1,200 milligrams. Perhaps this may be better for those patients who fall off the dosing scale. XMAB, no use in humans yet, but it has shown promise in mouse models over omalizumab, binding 40 times more. The next target that really is probably the next up for being a big player in the anti-IgE class is ligelizumab. It's an antibody that binds free IgE, significantly higher affinity than omalizumab. Again, it's important in that it may be able to dose patients with higher IgEs. It has shown a little bit greater efficacy than omalizumab in inhaled allergen challenges, so there's promise there. And there is actually a study of safety and efficacy in peanut allergy in development right now. So look forward to see what comes out of the IgE agents. The next target that's really come into the picture of food allergy over the last few years is IL-4, interleukin-4, and the main agent in that class is dupilumab. It's an antibody that binds the interleukin-4 receptor and prevents signaling by both IL-4 and 13. In the setting of food allergy, these guys are not the best news. They promote Th2 differentiation and drive mechanisms of allergic response up and down the pathway, including telling the B cells that they need to produce more IgE, leading to some peripheral and esophageal eosinophilia. It's already been approved for other atopic states. And so the thought is, let's start using this in food allergy, in which there's a lot of overlapping mechanisms. The potential benefits over anti-IgE agents is really the broader inflammatory pathway inhibition as you move up the cascade. We suspect that there may be a lower incidence and severity of adverse events, specifically GI, as well as potential impact on improving sustained unresponsiveness. So currently there are a few studies going on in food allergy. There are 2 phase 2s looking at peanut tolerance, one as its own agent, the other looking at it with peanut OIT in those aged 6 to 17. The MAGIC study is just gearing up. Near and dear to my heart, it is actually the study that I hold the IND for and I'm sponsor of here at Stanford. We're working with Mayo Clinic and Phoenix Children's in Arizona for this one. We're actually looking at milk tolerance following dupilumab treatment. Milk is often associated with a higher incidence of gastrointestinal adverse events. So we're hoping that dupilumab will really be able to see the reduction or prevention in those AEs that persisted when we use an agent like omalizumab. This will be in patients 4 to 50 years, and we're just opening up enrollment and recruitment as we speak. Last but not least, the combined study, also sponsored by Stanford and conducted with UCLA and UCSD. In this study, we're not only looking at dupilumab, but we're bringing omalizumab into the picture as well, combining the two for a multi-target strategy looking at different time points during therapy. We're really investigating pre-treatment with Xolair, which really does a good job at increasing the allergen threshold where the patient can start dosing, and then switching over to dupilumab when OIT begins, which we suspect might do a better job at suppressing some of those persistent AEs on board with Xolair. So I kind of alluded to it in an earlier slide, and I won't take too much time on the anti-interleukin 5s, but occasionally patients will develop EOE, eosinophilic esophagitis, while on OIT, causing basically their only option to be discontinuing therapy. It's marked by migration and recruitment of eosinophils to the esophagus. Really, there isn't much option at this time. Interleukin 5 is known to play a critical role in that process. So naturally, anti-interleukin 5 agents are being considered for pre and adjunct treatment during OIT to prevent this. Currently, there's no studies ongoing, but it's definitely in the pipeline. It's really only a matter of time in those who probably previously developed EOE and have food allergy. This may be a good place for the incorporation of these agents. If we can suppress the IL-5 and remove the EOE, we might be able to bring OIT and the benefit of it to these patients who experience EOE. Moving further up the cascade and the allergic pathways, co-stimulatory inhibitors are actually one of the most recent strategies currently under study. The way they work is a little more complex than the other ones discussed. But in broad terms, normally allergen-specific T and B cells require the simultaneous binding of two different sets of receptors, not just one, to survive and activate. I guess you can kind of think of it as an appliance that for some insane reason requires two plugs to work. It needs both to really function. First binding is the allergen piece being presented by the APC binding to the T cell or B cell receptor. The second is CD80 or 86 on the APC binding to CD28 on either cell. Luckily, adacept exists and is an agent that combines CD80 and 86 on the APC, preventing that second set of binding. And so when only that first half binds with the allergen, it's hypothesized that the T and B cell population will either die, become allergic or non-allergic, or switch down to a regulatory tolerogenic path. There is actually currently a phase 2A study, Atari, looking at this agent, adacept with peanut OIT, as well as sustained responsiveness after 12 weeks of discontinuation. As always, limitations. As we move up the cascade, like we said earlier, broader immune suppression may result in greater incidence of adverse drug reactions versus anti-IgE and IL-4 receptor alpha. And last is all the way at the top of the cascade, the anti-alarmins, namely IL-33, 25, and TSLP. These are located at the very beginning of development and maintenance of FA. Like others, the broader suppression further up may have advantages in promoting SU and preventing AEs. In mouse models, studies have shown a lot of promise, but so far, there's only the one study of these agents in this class. That's itokimab, antibody that binds IL-33. Did show promise in decreasing some of the factors that are driving allergies, but at this time, the manufacturer has chosen not to prioritize research in this disease state, instead shifting to atopic dermatitis and eosinophilic asthma. Same limitations as the others. As you move upstream, you're having more risk for adverse events due to the drug itself rather than the allergen. So, you know, all of these agents discussed, they all share a similar basic function in the treatment algorithm, which is providing a safer way to undergo repeated allergen exposure at escalating doses, and secondarily improving the odds of SU. And you know, these are all agents as well in this pathway. They're all being considered in the pipeline, but again, they're just a few. There's really been an upswing in the interest in finding biologic targets, incorporating them in food allergy. And I know we're over time, so I'll just go, you know, I'll just skim these two. As we talked about before, the modified allergens, you know, this is, I think, the next phase of food allergy therapy. It avoids the need of those biologics altogether if designed correctly. The allergens are designed to avoid recognition by mediators of allergic reactions, so no need for biologic suppression, with a gradual shift to T-reg dominant through the repeated exposure of naive T cells to the allergen. And I know I've talked about this one in previous talks, but just an update with the DNA vaccine that we have. This one codes for the peanut. It's taken up by APC, converted into peanut protein, and it gives repeated exposure to the T cells without ever entering the bloodstream or the serum. So you can avoid the reaction while guaranteeing exposure to the T cells and retraining. 2 phase 1 studies have really just wrapped up this, so fingers crossed we'll be moving into phase 2 and looking more efficacy. The next one in the pipeline is CMP 201. Same kind of thought process where you're shielding the allergen from the immune system, only this one uses nanoparticles to coat the peanut, shielding it from the immune system while still opening up where it needs to, exposing itself to the T cells, precluding allergic reaction. And phase 1 and 2 are starting in 2021. There's actually good progress on this one as well. And my last slides here, microbiome I have thrown in at the end. I'll leave this for you guys to look at on your own. As I've talked about before, current updates are there's really inconclusive data right now in support of probiotics for a number of reasons. Difficult to work with. There's just not enough known about what the whole microbiome does. You know, we know that it does have a link to food allergy, but we really don't know how best to utilize it for our own gain at this point. There's a number of ongoing studies. Notably, there is oral encapsulated fecal microbiota transplantation with or without antibiotics. Antibiotics used to flush out your existing bacterial population, giving the fecal microbiota a chance to take hold. And I think right now that's our best shot at getting a full array of strains that we need in order to make a big impact over just one strain. Some of you are cozied up at home asking, Andrew, how can we avoid more humans needing to eat feces? Which is a great question, which leads to our last slide. Primary prevention. So right now we're finally getting into studies looking at preventing food allergy in the first place. This is a study sponsored by Stanford with 3 additional sites in the US and 1 in the UK. Really looking at infants with atopic dermatitis evidence by 10 weeks of age. Can we implement a daily skin barrier repair routine that can stay off the effects of food allergy in the first place? And with that, I will turn it over. I'm over time, and Tina's yelling at me.
No, we're just a little bit over time, and I do apologize for those who were having some issues.
Yeah, but I'll leave up the slide. Leave up the slide for our resources. If anyone's interested in a snapshot of our current goings on, but feel free to check that out, and we'll open it up for questions.
Andrew, can you hear me?
Yeah.
Yeah. So if you want to open the Q&A session, there's 5 questions in there. And for those who cannot stay, and I'll send out the recording once it's done, and share this with you if you have to log off. So I'm going to hand this over to Andrew to answer some Q&As in the Q&A panel. The first question goes.
Yeah, where is it? Oh, I got it. Never mind.
Go ahead.
Yes. Okay. So I will go through these one by one. Magic study is actually starting any day now. We just finished the SIV with Mayo and Arizona. I have study drug in the pharmacy right now. And there really is no delay now. Medication's available. We're actually receiving it from Regeneron. We received a partnered grant with them. And so right now we are ready to roll. We're ready to start recruiting at our site. Let's see. Limitation on vaccinations while in a trial. Currently none. Really, you know, we've had this question many times throughout the pandemic, and we've done what we can to avoid needing to, you know, obviously we don't want anyone to delay their COVID vaccines or flu shots. At this time, we really haven't had any interactions with the COVID vaccine or flu shots in terms of recruitment or needing to change your dosing. They can all be used at the same time because they work on distinct parts of the immune system that are completely separate from each other. Kind of like we talked about with the antibodies being broad versus specific. Right now, we're really just using Anti-IgE and dupilumab, which are more specific to the allergen pathway than your entire immune system as a whole. Let's see, latest on the use of Dupixent and Milco-IT. There are trials right now, but the MAGIC study will be the first on Milco-IT and Dupixent. Just to date, it's really just been peanut. Peanut's always the big focus and the first one that a lot of these industries go to when they're bringing an antibody into study. So we kind of wrote it a little bit different. We went with milk. Got to show those with milk allergy some love. And I think in this disease state where adverse events in the GI system are a little bit more prevalent, dupilumab might have a bigger role here. And I think we'll be able to see the difference between normal dupilumab a little bit better. The evidence that biologics and immunotherapy could improve sustainable response. Right now, you know, this one is a little bit complex. You know, the basic OIT, where we're just repeatedly exposing the T cells to allergen, we're finding that, you know, on the surface level, you're retraining, but there are populations of B cells and T cells that are long-lived. And after you stop dosing, it seems that they're still living. And this is deep down into the bone marrow. And so by using a biologic, we're hoping that we can kind of augment what's already happening if we downregulate some of the cytokines and the other players in the pathway, on top of just the repeated exposure and what that repeated exposure does. We're hoping that we can make a bit more robust, you know, cell death of the memory B cell and T cell population as a whole, and more suppression in the swing from Treg or Th2 to Treg. Negatives of dupilumab, there, you know, for the most part, there aren't too many. The only limit would be that it's a little bit more broad than anti-IgE. You know, now instead of blocking IgE at the end, which is really specific to food allergies, dupilumab blocks 4 and 13. 4 and 13 have a few roles in the body, obviously, you know, it's not all bad, but for the most part, it's still pretty limited to that inflammatory pathway. And it already has approval for a number of other atopic diseases. And so the safety's, you know, pretty well known. Typical duration of OIT for successful treating of allergy. OIT on its own, we've seen anywhere from a year to 3 years, depending on how high the patient wants to get. I'd say on average, it usually takes about a year, year and a half to get up to a nice maintenance dose safely. And so without the biologic, you're looking at a long-term single for just one allergen. When we incorporate the biologics, we've seen, especially for something like Xolair, some patients are able to max out in a number of months to all of their allergens, not just one. Again, that's highly variable between patients. We've seen a group of patients that just don't react very well at all. And that's kind of what we're trying to analyze right now in our own patient population. Hopefully we have some more answers next time around. Side effects of Xolair, especially impact on children's growth. So far, nothing has really been seen. We haven't seen any impact on growth. And I think that's in thanks part to its specificity for just IgE. Going into a little more detail, I've always said IgE, it's all specific to allergy. It does have one other role, and that is in the prevention of parasite infection and Hellman's infection. But most of our patients are not trekking through the jungle during treatment. So I think that's a risk everybody's willing to take, especially because it's a temporary use. Usually we only use Xolair for about 8 to 16 weeks. The NIH study where they're looking at it for a full year is the longest duration we've seen, but it's not unheard of. I mean, people with allergic asthma will be on Xolair because they're not desensitizing. They're just using it to suppress the allergy itself indefinitely. And so far, no evidence that it affects growth, given that IgE itself does not play a role. We know so far. That could always change. Does successful OIT with a biologic for one allergen help with success for future OIT with other allergens, lasting benefit or accelerating future process? Typically, we haven't really investigated it that much because of how long an allergen takes. By the time the next allergen rolls around, it's usually, we're not following the patient anymore because they're so far out, they're not doing OIT again. OIT with a biologic for one allergen, the biologic itself is non-specific. So if it works for one, it works for them all. But if you were to discontinue the biologic and, it kind of washes out, you'll still have your other allergen. We don't really have that much data on the benefit or acceleration of future progress. Hypothetically, if the other allergies still exist, you're still in an inflammatory state, even though you're tolerating the one allergen. And so I think, you're definitely going to probably want a biologic on board, or you're going to want to go through the process for the next allergens the same way you would for the first time around. But we don't really have that full data yet. It'd be definitely interesting to look at. And it's something that we're always banking samples for to try and understand these questions. How does fixing one move on to the next? Any other questions? I know there's a lot of info to go through.
Out of time, but also finished with all the questions. Thank you everyone for sticking around the extra 20 minutes after. I will definitely send this recording out to anybody who missed the beginning, couldn't see the screen, or just wants to see and hear Andrew talk again. Thank you again.
I know, my lovely swooning voice. Absolutely. And if anybody has any other questions, make sure you submit them, and I'll do what I can to get you an answer.
Yeah, thank you so much, Andrew, and thank you everybody else. Have a good day.
All right.